Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjong classes. Dear student, we are in a continuation of the chapter of class 11th, that is photosynthesis in higher plants. Till now, we have already talked about the mechanism of light reaction and dark reaction, which are important to understand the photosynthesis that we have covered. We have covered cyclic, non-cyclic photophosphorylation, C3, C4, cam cycle, all these things we have discussed. Now we are remaining with the photorespiration and the factors affecting photosynthesis. So now we are going to talk about the photorespiration first. So photorespiration, okay. Respiration means obviously energy gaining mechanism, we can say, or the use of oxygen. And photo means the presence of light. Because of this, we are calling it the photorespiration. So here to understand photorespiration, we must know a little more about the first step of Kelvin pathway, the first CO2 fixation step, which we have discussed in the C3 pathway. And we know that this step is particularly coordinated by the enzyme or the enzyme involved in this first step, that is a Rubisco, that is the ribulose 1,5-biscoacid carboxylase. And we have already studied that Rubisco has a dual nature. Either it can bind with O2 or either it can bind with CO2. If it binds with CO2, it will act as carboxylase. If it binds with O2, it will act as oxygenase. When it binds with the CO2 and act as carboxylase, the C3 cycle takes place in C3 plant. But when this Rubisco bind with O2 and act as oxygenase, the photorespiration takes place. So this is the reaction where RUBP combines with CO2 to form a two molecule of 3 PGA that are catalyzed by Rubisco in the C3 cycle. But Rubisco is the most abundant enzyme in the world, and it shows that it has a dual activity. It can act as a carboxylase, it can act as oxygenase. If it acts as a carboxylase, it bind with CO2, and if it acts as oxygenase, it bind with O2. Okay. So when it acts as oxygenase, photorespiration occur. So what we will write? When Rubisco function as oxygenase and Rubisco act as oxygenase, photorespiration take place. Okay, that is the thing. Now this photorespiration, if we talk, this photorespiration is also called as number one C2 cycle, number two CO cycle. PCO stands for photosynthetic carbon oxidation cycle. Third, glycolate metabolism. Okay, these all are the names of the photorespiration. And it is considered that this cycle of photorespiration was discovered by Decker and Teo in Tobago. That is a C3 plant. Okay, so that is the thing. So photorespiration is a characteristic of a C3 plant. Now here, to carry out a photorespiration, three organelles are involved, and these three organelles are number one chloroplast, number two peroxisome, and number three, that is mitochondria. These three organelles are involved to carry out a photorespiration. Now, how this photorespiration takes place or cycle takes place, that we have to understand, and this is the cycle mechanism of cycle. Okay, or in this, the first organelle which is involved, that is a chloroplast, then peroxisome, then mitochondria. All these three are coordinated with each other, and they are involved to carry out this photorespiration. Now, how this photorespiration takes place, that we know. Here, as we know, this rubisco act as oxygenase and react with RUP. Oxygen react with RUP. Now, RUP, we know that it is of a 5 carbon. Okay, it is of 5 carbon. And along with this RUP 5 carbon, oxygen has no carbon. Now, here we take a 2 molecule of RUP and 2 O2 molecule. Then this RUP 5 carbon it break into the 2 carbon compound and 3 carbon compound. But obviously, this is the 2 molecule if we are taking, then 2 molecule of phosphoglycol it will be there, and 2 molecule of phosphoglyceric acid will be there. That is a 3 phosphoglyceric acid. So 2 molecules of 3 phosphoglyceric acid remain here in chloroplast, but 2 phosphoglycolate move further. Now, out of this 2 phosphoglycolate molecule, 2 phosphoric acid, from each molecule 1 phosphoric acid or inorganic phosphate get removed. So 2 inorganic phosphate are removed and 2 glycolate molecule are obtained. Now, these 2 glycolate molecule pass on to the peroxisome from chloroplast. Here, oxidase enzyme will occur, that is the glycolate oxidase enzyme, which is known as marker enzyme. That will carry out oxidation, and 2 molecule of glyoxylate will be obtained, and along with 2H2O2 will be obtained. Now, 2 molecule of this glyoxylate, further using a amino group from this glutamate or glutamic acid, they get converted into amino acid, that is glycine. But here also 2 molecules of glycine are there, and it is amino acid. Now, what happens to this 2H2O2? Here, catalase enzyme will work, and catalase enzyme break it into the 2H2O plus O2. Okay. Further, this 2 glycine molecule of 2 amino acids, because it has accepted amino group from the glutamic acid. This 2 glycine molecule, which is of 2 carbon, it is of 2 carbon amino acid. So total 4 carbon is there. These all molecules are of 2 carbon. We remember. Okay. So this 2 molecule of glycine, and each glycine has a 2 carbon. So total 4 carbons are there. Out of 4 carbon, a 1 molecule of serine is getting synthesized, and serine is also amino acid that is of 3 carbon. So out of 4 carbon, 1 carbon is losing. Because of this, the 3 carbon serine is obtained in a mitochondria. Here, along with the decarboxylation, deamination also occurs. Amino group also get released, and along with this, NAD get reduced into the NADH. This CO2 is released outside. So here, oxygen is using and CO2 is releasing. Now, serine again come back to the peroxisome. Here, it releases amino group from alpha ketoglutarate acid. So it releases one amino group here, okay, and suppose 2 molecule of glutamic acid are there, alpha ketoglutarate acid, then it can synthesize a 2 glutamate molecule. Now it receives one amino group from this one amino acid of 3 carbon and another amino group, that is this one. This amino group will come and react here with the 2 alpha ketoglutarate acid. So with 2 alpha ketoglutarate acid, 2 molecule of amino group, one from the serine and one from this one, get reacted and 2 glutamic acid are found. 2 glutamic acid when break into 2 alpha ketoglutarate acid, it will release 2 amino groups that are accepted by glycosylate and converted into glycine. Now here, with the release of amino group, serine get converted into one molecule of hydroxypyruvate. This is of 3 carbon. Here NADH get converted into NAD, and here you will see the one molecule of glycerate that is also of 3 carbon. This glycerate passes to the chloroplast again. Here one ATP is used, and ATP, with this use of ATP, conversion of ATP into ADP, one phosphate is released by glycerate and that converted into 3 phospholipid glyceric acid that is of 3 carbon, and this is one molecule. Now this one molecule along with this 2 molecule undergoes C3 cycle, and this C3 cycle will regenerate 2 molecules of RUP, because in C3 cycle we know there is a regeneration step. So this way the whole C2 cycle carry out. Now you can see on this side, right hand side, you can see that as a 2 carbon molecules are moving from chloroplast to peroxisome to the mitochondria, and 2 molecules are there, means 4 carbon are passing. And when we go back, here you can see the serine Hydroxy, they all are of three carbon. So three carbons are coming back. Yes or no? We can see here. So here, after understanding this photorespiration, we can say that the one CO2 is releasing here. Because of this, we can say an important statement that approximately 25% CO2. You can see four carbon coming and three carbon. So one carbon is lost. So 25% CO2 or carbon is lost, and 75% carbon is recovered back. So this thing occur in this particular photorespiration. Now one more thing you can see that here O2 is using, CO2 is releasing. There is no synthesis of ATP, and because respiration we are calling, then also there is no synthesis of ATP, and in spite of no synthesis of ATP, one ATP is getting used. So it is we are calling the respiration, but there is no benefit from this. So because of this, it is considered that photorespiration is a wasteful process. Photorespiration is a wasteful process. Is a process because there is neither synthesis of sugar, there is neither synthesis of sugar nor of ATP and NADPH. Rather, it result in the release of CO2 with the utilization of ATP. So rather it result in release of CO2 with the utilization of ATP. Because of this, we are calling it as a wasteful process. So this is what we can say the photorespiration. Now, what are the conditions which favor the photorespiration that we have to understand? So first we talk about conditions that favor photorespiration. So if we talk about the conditions which favor photorespiration, okay, then the first condition is high light intensity. It is known that it is a main condition for photorespiration. For photorespiration, because light is obviously required, and it is responsible for the second and third condition. Second condition is higher concentration of O2 and lower concentration of CO2. And lower concentration of CO2 inside mesophyll cell. So there is a higher concentration of O2 and lower concentration of CO2 inside a mesophyll cell. And under such condition, rubies bind with O2 and act as oxygenase. Under such condition, rubies bind with O2 and act as oxygenase enzyme. Okay, and the third condition. So this is the condition where a Rubisco will act as oxygenate and carry out a photorespiration. Third condition, as I told you, that is a high temperature. That also particularly favor the photorespiration. So this availability of oxygen reduces the rate of photosynthesis, that is known as Warburg effect. So it is described by Warburg. So it is known as Warburg effect. So what is it? It is the decrease in, it is the decrease in rate of photosynthesis. It is the decrease in rate of photosynthesis by high oxygen concentration. So here oxygen is a competitive inhibitor, is competitive inhibitor of the carbon dioxide fixation, of the carbon dioxide fixation by Rubisco. So oxygen promote photorespiration, which reduces photosynthetic output. Okay, so that is the what we can say the photorespiration. Okay, dear students. So this is all about what photorespiration that we have understood very well. So what we have understood by studying this photorespiration, that Rubisco has a greater affinity to CO2 than the O2. And the binding is obviously competitive, which has a higher concentration that will bind with the Rubisco. If it is the relative concentration of O2 and CO2 that determine that which of the two will bind with the enzyme that we have discussed in C3 plant. Some O2 does bind to Rubisco and hence CO2 fixation is decreased, and so photorespiration can occur in C3 plant. Here the RUP, instead of being converted into two molecules of PGA as in the C3, it bind with the O2 and molecule form one molecule of phosphoglycerate. Phosphoglycerate is of two carbon, because of this we call it C2 cycle, and the phosphoglycolate, no sorry, phosphoglycerate is of three carbon and phosphoglycolate is of two carbon. So because of the phosphoglycolate is known as C2 pathway. In the photorespiratory pathway there is a synthesis neither of sugar nor of ATP. Rather, there is a release of CO2 with the relation of ATP. In a photorespiration pathway, there is no synthesis of ATP and NADPH. Therefore, photorespiration is restricted process that we have discussed in C4 plant. Photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. Means near about the Rubisco. In bundle cell, they have a capacity to increase the concentration due to the breakdown of malic acid. This takes place when the C4 acid, that is malic acid from the mesophyll cell, is broken down into bundle cell and release CO2, resulting in increasing the intracellular concentration of CO2 in bundle cell. That's why Rubisco will act as only carboxylase. In turn, this ensures that Rubisco function as a carboxylase, minimizing the oxygenase activity that we have discussed in C4 plant. They lack a photorespiration, and in addition, these plants tolerate high temperature also. So now, after studying this photorespiration, if we compare the C3 C4 plants, then with some characteristic we can compare the C3 and C4 plant, which are important from. So cell type in which the Kelvin cycle takes place. So we know that the Kelvin cycle or C3 cycle takes place in C3 plant in case of the mesophyll cell, whereas in C4 plant it is in the bundle cell. Okay, so we have to choose from it. It is NCERT table. Cell type in which the initial CO2 fixation occurs, so mesophyll cell in which first CO2 fixation occurs, and in C4 plant also it is a mesophyll cell. How many photosynthetic cells types does the leaf have that fix CO2? So we know that only one type of cells are involved in C3 plant, that is a mesophyll cell, whereas the two type of cells are involved, bundle sheath cell and mesophyll cell in case of the C4 plant. Then if we talk about other characteristics between the C3 and C4 plant, next characteristic is which of the primary CO2 acceptor in C3 plant. So RUP, a 5 carbon compound, is a first CO2 acceptor, and in PP, a 3 carbon compound, that is a first CO2 acceptor in C4 plant. Number of carbon in the primary CO2 acceptor, number of carbon in RUP is 5 and PEP is 3. So 5 and 3, the number of carbon in primary CO2 acceptor. Which is the primary CO2 fixation product? The first product which is found by the primary CO2 fixation by the Rubisco peptidase, that is PGA in C3 plant, and which is 3 carbon, and oxaloacetic acid, which is a 4 carbon in the C4 plant. Now, due to the PGA, which is a 3 carbon, this C3 cycle is known as C3 cycle, and due to the formation of first stable product, that is the oxaloacetic acid of 4 carbon, because of it we call it C4 cycle. Number of carbon in the primary CO2 fixation product, that is the 3 and 4, because of this they are named. Next characteristic, does the plant have a Rubisco? Yes, C3 plant have and C4 plant also have, but in C3 plant it is present in mesophyll cell, and in C4 plant it is present in bundle sheath cell. Does the plant have peptidase? No, in C3 plant you will not see the presence of peptidase, but in C4 plant mesophyll cell you will see the presence of peptidase enzyme, which carry out the carboxylation of PP and formation of oxaloacetic acid. Which cells in the plant has Rubisco? Obviously mesophyll cell in C3 plant and bundle sheath cell in the C4 plant. CO2 fixation under high light intensity, medium in case of C3, but C4 plant can tolerate high light intensity. So C4 plant, the at high light intensity they survive. Next characteristic is whether the photorespiration is present at low light intensity. So photorespiration can occur in case of the C3 plant, but negligible in case of C4 plant, or absent in case of the C4 plant. Whether photorespiration is high light intensity, yes, it is present in high light intensity C3 plant and it is negligible. So at a low light or high light, photorespiration can occur in case of the C3 plant. Whether photorespiration could be present at a low CO2 concentration, low CO2 concentration, the C3 plant will perform a photorespiration because the oxygen concentration will increase, but due to the decrease in CO2 concentration in C4 plant, the photorespiration cannot occur. So negligible is there. Next characteristic, whether photorespiration could be present at high CO2 concentration. High CO2 concentration is there, then photorespiration cannot occur in both C3 and C4 plant. Temperature optimum for C3 plant is 20 to 25 degrees Celsius and C4 plant is 30 to 40 degrees Celsius. Examples of the C3 plant is sunflower, sugarcane, and other examples already have told in the while studying the C3 and C4 plant. Okay, so this is the what we can say the thing. Apart from being remembered in examples, we should remember one some more example like wheat and rice. In C3 you should remember, and here the maize, sugarcane, and sorghum. These are the important examples that you should remember in the C4 and C3 plant. Okay, so that is all about the photorespiration. So RUP carboxylase or Rubisco, we can act as a RUP oxygenase at a low CO2 concentration and high oxygen concentration. So low and high become the correct answer. So now we are going to talk about the factors affecting photosynthesis. So we have completed here photorespiration. I hope you are now thorough with the photorespiration. That is a C2 cycle. Okay. Now we will discuss the factors affecting photosynthesis in the separate lecture. Okay. Thank you everyone. I hope you understood photorespiration very well. Thank you.